Bismillah ar-Rahman ar-Rahim. We continue of genetic polymorphism, and as I told you last period, we have to take, or we will take the DNA from a normal part. Look to this. In order to solve this problem, in order to solve this question, we take the DNA of a normal, one normal cell from any normal part of this individual. We cut them using the same restriction enzyme. Now the problem becomes what? Becomes to know what is the number and the size of the fragments obtained with each DNA. If we obtain the same number of fragments and the same size of the fragments, the same size, no difference, we say that the two DNA are identical. If not, so there is genetic polymorphism. So how can we know the number and the size of the fragments? In reality, in order to find the number of the fragments, we have to make a technique called gel electrophoresis. Gel electrophoresis, in order what? To separate the fragments and to analyze the size of the fragments. In order to understand this technique, I will draw with you, okay, the experimental setup, and I will explain on our drawing. So please draw in same time. So as you see here, we get a container. I am drawing a container, please draw it in same time. And what? This container presents cavities. I am drawing now the cavities. Okay, so we have here a container presenting cavities, 3 cavities I have here in this container. And what else? In reality, this container is connected to an electric current, to an electric field, to a battery or to electric generator, a battery, for example, car battery. Look to this. Here we have the negative pole and the positive pole. Look to this connection, please. This is the battery, this is a generator, electric current, and we have connecting this electric current to the container, to the recipient or container, such that the positive, the positive size, the positive pole is here. This is the positive pole of the battery or of the electric generator, and the second one is the negative pole. Then what? Inside this container, there is a jelly substance. We say it is gellose or agarose. It is a jelly. Sorry. It is a jelly substance. It is the gellose or agarose. جلو عادي بكل بساطة. فإذا هيدا container, it presents cavities, okay, and it's connected to an electric current. It contains, this container is filled with gellose or agarose. I mean by this a semi-fluid, semi-solid or semi-liquid, يعني not liquid and not solid substance, jello العادي. Then what? In this, we will put the restriction fragments and the fragments of the obtained restriction enzyme to the DNA. So here we have the restriction fragments. We put in these cavities the restriction fragments obtained. As you know, we added to the DNA the same restriction enzyme, and once we obtain the fragments of DNA, called also restriction fragments. Restriction fragments means the fragments of DNA, and DNA fragments. We name them also restriction fragments. So in this cavity, we put, for example, the restriction fragments of DNA 1, if you want, DNA 1, and the second DNA 2, DNA 1. and of the normal cell, for example. And here we have the DNA too. We want to compare the number of fragments and the size of the fragments obtained, okay, once we added the same restriction enzyme to each of these two molecules. So the fragments obtained are put on this cavity, and now what? In reality now, since the DNA has a global charge, the DNA, because of the presence of the phosphate PO4 minus minus minus, phosphoric acid, phosphate function, it will have what? A negative charge. So have a global negative charge, and they will be attracted. So the fragments of DNA will be attracted towards what? Towards the positive pole of the electric current. Good. So what? DNA contains phosphoric acid, group phosphate, PO4 minus minus minus. It means that it has global negative charge, and the fragments will be attracted toward the bottom of the container, because in the bottom we have the positive pole of the battery is connected to the bottom of the container. Good. Now we have migration. Now we have migration of the fragments downward in this direction, like this. So I will write here direction of the migration. Please draw in same time. Direction of the migration. Migration direction. Yani movement, direction of the movement of the fragments of DNA will be downward, because the bottom of the container is connected to a positive pole of the battery, to the positive pole of the battery. Good. So now what? Why we use glucose or agarose esters? No, if we put water, liquid water, in this case the fragments will go anywhere randomly in the liquid. We will lose them. If we put a solid substance, there will be no migration in the solid substance. So we added glucose, or we put glucose in this container in order to let the movement occur, and no losing of the fragments in all directions. So they will move downward, and this movement or this attraction will be based on their size. Okay, yes, it is. As a magnet، خليك تخيل معك مغناطيس، وعندك مسامير صغار كتير ومسامير أكبر ومسامير أكبر. إذا قربت المغناطيس عليهم أكتر شيء مين راح يجذب لعنده؟ طبعا الصغار كتير دغري راح ينطوا عليه. فإذا more the fragment has less size, more it will go downward. بينما فوق راح يبقوا الفراجمنتس اللي عندهم size أكبر. And what else? This container is graduated. Okay, look to this. Look, please. Here we have graduations. مثل المسطرة graduations. بس كيف graduations موجودة هون؟ يعني look to this. Here we have, for example, 500 base pairs. The size of the fragment is measured in not kilometer, ولا المتر, ولا centimeter, ولا base pairs. You know this. So, and تحت مثلا 400 base pairs, 300 base pairs, 200 base pairs, 100 base pairs. Now what? Now these fragments will move downward based on their size. طيب خلينا نفترض انه I obtained this, for example, here, and what here, for example, another fragment here, for example, and for example كمان fragment here. طيب فإذا we obtained now 3 fragments, and look to the level. We can know the size of each fragment by looking to the level they are on it. Good, but it says now, are we able to observe, to see these fragments of the molecules? طبعا لا. أكيد لا. we cannot see molecules. How can we let these fragments visible? First of all, we add to them a substance. We add to the fragments substance called ethidium. Ethidium bromide. Ethidium bromide. This substance, once we added it to the gels or on the fragments, they become visible when we expose them to ultraviolet radiation. So ethidium bromide plus ultraviolet radiation, so the fragments become what? Visible. Let me write this remark. Ethidium bromide to let fragment be visible once exposed to ultraviolet radiation. Good. So let us make a general view on this, on what we have on board. We have a container. In this container we have gels or agarose. It is a jelly substance that is not solid and not liquid, and we have cavities where we will put the restriction fragments here in them. And this gels this container is connected to an electric field, such this, plus in the bottom and minus upward. Now these fragments, since DNA contains the phosphoric acid, it means contain global negative charge, implies that the attraction will happen toward what? Toward positive pole of the battery. These fragments are attracted according to their size. More the fragment has less size, more it will go downward. We separated like this the fragments based on their sizes. This container is graduated, وبالتالي once we added ethidium bromide and we expose the fragments to ultraviolet radiations, we can see them, we can know how many fragments we have and what of each fragment. Now, as you see, the DNA one, the first DNA is cut by the restriction enzyme into 3 fragments. بالحقيقة بيكون في عندي hundreds or thousands of fragments، مش 3 بس، ما عندي وقت أنا أرسم 10,000 fragment مثلا عم بحكي. طيب، فإذا هون واضح إنه we simplified the drawing. 3 fragments, 1 fragment of size having a size of 500 base pairs. This fragment here has a size of 250 base pairs, and this one 200 base pairs. اوكي استاذ. طيب وشو كمان بعد اذا بدك تعطينا ملاحظه صغيره. Look to this. What we obtained here in reality, we name it a restriction map. We obtained here a restriction map. We'll name this. This is called restriction map. All this, we name it restriction map. All this, all the fragments that are now arranged like this, they form what we name. So this is what we name a restriction map of the individual. Restriction map, like this. Restriction map. Okay, Ustaz. طيب, now what? If I am telling you, if the other DNA gives us the same number of fragments and the same levels, it means what? These two DNA are identical and there is no genetic polymorphism. And if there is any difference in the number of fragments or the size of the fragments, we say that these two DNA are different and there is genetic polymorphism. Good. And what? Another idea. I want you to tell you this. This technique is what we name gel, gel, because we are using gels. Electrophoresis, because we are using what? Electric current. So gel, electrophoresis. 1 and 2. Gels and electric current. Gel electrophoresis. It is the gel electrophoresis. It is the technique we use to, the procedure, if you want, we use to separate the restriction fragments and to recognize the size of these fragments. Okay, RAM at the end, RAM. And before writing this RAM, suppose we have a crime. Please pay attention for this. خلينا نفترض انه there is a crime and هيدي المرة اللي ماتت كان في تحت ضفيرة جلفته للمجرم. حرام المجرم جلفته. اوكي. وعندنا 10 suspects. Okay. How can we know which one is the criminal? right. عندنا امرأة لقيناها ميتة وقبل ما تموت كان جلفته للمجرم. اوكي، يعني في تحت ضفيرة خلايا من المجرم. وعندنا 10 suspect, 10 individuals we suspect them, one of them is the criminal. How can we know which one is the criminal? Okay, so it's important. قبل ما اسأل هيدا السؤال خليني اكتب جملة وانتو تكفوا لي اياها. Please. Two different individuals have 3 نقط restriction maps. 3 نقط. 2 different individuals have what? Have different restriction maps. لأنه when you say 2 different individuals يعني 2 individuals having different DNA يا خيي. يعني لما تعمل restriction map للأول و restriction map للثاني على الأكيد you must obtain 2 different restriction maps لأنه DNA are different, so we will obtain different number of fragments and different size. نرجع هلا نرجع للكريمينال. انه مرة ماتت وفي تحت ضفيرة خلايا من المجرم. اوكي، حال كيف نحن رح نعرف من suspect 10 suspects which one is the criminal? That's it. نعمل restriction map للسلسلة اللي تحت ضفير المرة و restriction map لكل واحد من 10 suspects. اللي بيطلع الريستريكشن ماب تبعه مثل الريستريكشن ماب تبعت الساس تحت ضفير هيدي المرأة، يعني بيطلعوا ايدنتيكال، بيكون he is the criminal. That's it. Very good. That's it. Okay. Now, in order to finish this study, I want to return to our PowerPoint presentation. As you see, this is our document. Sorry for the French words. And this is the restriction map. We add to them ethylium bromide to the fragments become visible as I told you. And this is the direction of the migration. Smaller fragments are downward at the bottom, near to the bottom, while the fragments having bigger sizes will become near to the surface. Okay, look to this. This is called a restriction map, the restriction map of the individual. Okay, this represents the visualization. It means when we let the fragments become visible using ethidium bromide and what and ultraviolet radiation. Look to the fragments. Here are the restriction fragments. Okay, the restriction we can count them, etc., etc. So as you see, this is the restriction map. Yes, we can see it under under ultraviolet radiation once we added ethidium bromide. Yes. Good. Now to finish our study on this document, there is a notion to understand this document. Please, we have 5 individuals, individual A, B, C, D, and E. We are there with the restriction map. Okay, this is the restriction map of Tiani. So, do we have identical restriction map here? No, and what individuals that are true twins. Now, why this? Another individual. This is what we name length polymorphism, because they do not have all the same size, so we have different types of restriction maps and different levels of the fragments. That's it, and I am waiting for your questions.